This is a Journal Club presentation for our recent publication, which was a group effort between co-first authors Preston Kroll and Jonathan Fox, several members of the Goldstein lab at UCLA, colleagues at UCLA, and collaborators at UT Southwestern. As the senior author, I will describe what we did and what we found in this study. As we age, many tissues lose mass. Progenitor activity, which is consistent with the capacity to regenerate, is lost with age in many tissues. Aging in the prostate, however, is associated with growth-related diseases like prostate cancer and benign prostatic hyperplasia, or BPH. Risk of cancer and BPH increases with age, and a major unanswered question is, put simply, why? What happens to the prostate during aging? To address this question, we decided to use mice because we could follow genetically identical animals over time. We used 3-month-old mice, which are considered young adult, and compared them to 2-year-old mice, which are roughly equivalent to 80-year-old men. We first noticed that old prostates were heavier and contained many more cells than young adult prostates. The prostate is a highly branched structure, like a tree, so we asked whether the prostate becomes more branched with age. However, we did not find a significant difference in the number of branch points in any of the lobes of the mouse prostate as a result of aging. We then asked what kinds of cells are increased in old prostates. The prostate contains an inner layer of luminal cells, surrounded by a layer of basal cells, with stromal cells, and what we refer to as Lin-positive cells, comprising the immune cells and endothelial or blood-lining cells. The results were very clear that there's a dramatic increase in Lin-positive cells in the old mouse prostate. We confirmed that this was caused by an increase in immune or inflammatory cells by showing a significantly higher percentage of CD45 positive cells in old prostates. So, old mouse prostates are heavier, they contain more cells, specifically immune or inflammatory cells, which led us to ask, what kinds of immune cells are found in old mouse prostates? To address this question, we used a technology called mass cytometry, or CyTOF, where cells are stained with antibodies conjugated to heavy metals, and single cells are analyzed using a mass spectrometer. This allows for a large number of markers to be evaluated simultaneously. We then used algorithms including t-SNE, or T-distributed stochastic neighbor embedding, which is a dimensionality reduction tool, in order to visualize complex data in 2 dimensions. In t-SNE plots, cells with similar features are clustered together. We can look at individual marker expression superimposed onto these 2D plots to highlight the CD3 positive T cells, the CD19 positive B cells, and the myeloid component. When we then show all of the immune cells from young adult and old prostates, it becomes clear that there's an enrichment for T and B cells with age. We can also quantify this result to show an increase in T and B cells in the old mouse prostate immune compartment. So we found an age-related shift in the immune compartment of the prostate with more T and B lymphocytes, and we wondered how these changes might influence the basal and luminal cells in the epithelium. Importantly, we could now ask this major unanswered question of whether progenitor activity is lost or maintained with age in the prostate. We decided to use the prostate organoid assay to measure progenitor activity, where cells are isolated from prostates, plated into a 3D matrix, and a subset of these cells will go on to form prostate-like organoid structures. We found that basal cells could form organoids at a similar rate, and this was retained when we passaged organoids into secondary culture, so we concluded that there's no loss of basal cell progenitor activity with age. When we looked at the organoids and measured their diameter, we found no significant difference in their size. We then evaluated luminal cells and found no difference in their capacity to form organoids in primary or secondary culture, demonstrating no loss of luminal cell progenitor activity with age. When we looked at the organoids and measured their size, we found that old luminal cells generate larger organoids on average. So we've begun to address this fundamental question, and our results suggest that progenitor activity is maintained in the old prostate. We also found that old luminal cells generate larger organoids, which led us to ask, what has changed within the luminal population? We performed RNA sequencing to measure gene expression in adult and old luminal cells. By highlighting genes that changed significantly with age, it became clear that many progenitor genes are elevated in old luminal cells. We used gene set enrichment analysis to show that the old luminal signature significantly overlaps with our previously reported luminal progenitor signature from human prostate. We selected one of these progenitor genes, called TROP2, and measured protein expression using flow cytometry, finding a significant increase in mean fluorescent intensity on old luminal cells. So we asked if progenitor genes like TROP2 get activated in old luminal cells, or if there's an age-related expansion of a pre-existing luminal population with progenitor features. We looked closely at TROP2 expression in young adult prostates and found a minor population of TROP2 positive luminal cells and compared them to TROP2 negative luminal cells. More of the TROP2 positive luminal cells were proliferating based on Ki67 staining. TROP2 positive luminal cells also formed organoids at a higher rate, and TROP2 positive luminal cells formed larger organoids. Most of the luminal-derived organoids contained 1 or 2 layers of cells, with high expression of the luminal marker keratin 8 and low expression of the basal marker p63. These findings suggest that TROP2 positive luminal cells are progenitors. Importantly, we found a significant increase in the percentage of luminal cells that are TROP2 positive in old mouse prostates. We asked, where are the TROP2 positive luminal cells located? In addition to localization in the proximal region, which we'd previously reported, we also found TROP2 positive luminal cells at sites of protrusion in the middle of ducts. We profiled gene expression from adult and old TROP2 positive and TROP2 negative luminal cells and found that these cells retain their signature with age. PSCA, or prostate stem cell antigen, is expressed in TROP2 positive luminal cells, regardless of age, and we'll come back to this marker later. So our results fit the second model, that there's an age-related expansion of a pre-existing luminal population with progenitor features. But we knew it was essential to determine if old TROP2 positive luminal cells retain their progenitor activity. TROP2 positive luminal cells formed larger organoids regardless of their age, suggesting that this capacity is inherent to these cells. Old TROP2 positive luminal cells formed organoids at a similar rate. In contrast, old TROP2 negative luminal cells had a reduced organoid forming capacity. Gene expression profiling showed a downregulation of cell cycle-related genes in old TROP2 negative luminal cells. So these cells are not just expanded with age, but they retain their progenitor activity with age, while TROP2 negative luminal cells lose this activity. When we calculate the actual number of luminal organoids formed from either TROP2 positive or TROP2 negative luminal cells, factoring in the frequency of each population and their organoid forming rates, we find that the vast majority of luminal organoids in young adult prostates come from TROP2 negative luminal cells. But then there's a major shift with age as the majority of luminal organoids in old prostates come from TROP2 positive luminal cells. Finally, we wanted to know, are these results in the mouse consistent with aging in the human prostate? We previously showed that PSCA is a marker of luminal progenitor cells in human prostate, and we found an age-related increase in these cells in human prostate. We also found an age-related increase in CD45 positive inflammatory or immune cells, similar to what we observed in old mouse prostates. Luminal progenitors were increased in BPH tissues compared to normal human prostate, and the same was true for inflammatory cells. Therefore, expansion of luminal progenitor cells is a conserved feature of aging in the prostate. When we look at the different cell types in the prostate, we see an increase in luminal progenitor cells and inflammatory cells with age in mouse prostate. This was similar to what we found in human prostates. As we age, many tissues lose mass, and progenitor activity, or the capacity to regenerate, is lost with age in many tissues. Aging in the prostate, however, is associated with growth-related diseases, and we can now show that progenitor activity is maintained with age in the prostate, which is caused, at least in part, by an age-related expansion of luminal progenitor cells. We previously found that luminal progenitor cells can initiate human prostate cancer in response to oncogenic transformation, which means that there's an age-related increase in the cells that are capable of or at risk for developing into cancer. We hope you enjoyed learning about our research. You can check out the paper on our lab website or the Cell Reports website. Thank you for watching this Journal Club presentation.